So exam Wednesday. So we'll do a review on Monday. So today's the last day for new material for this exam. We were talking last night about it at the review session. So it's just chapters 17 and 18 plus whatever we cover today, which is going to be a little bit in 19. So review on Monday. The lab meets next week. So you need to have finished labs 6, 6, and 7, right? You completed labs 6 and 7 so we can continue on. Next week what we're going to do is we're going to do a metal affinity chromatography purification of your protein, which is, I think it's fun. The resin's pink. You get a little bit of color in there. But and then we said the lab report will be due on Sunday. Not this Sunday, the next Sunday. All right, questions? Things we're doing? Okay. Then I was pulling pictures from a previous set of notes, and I thought this was interesting. I'd missed this. And so this is from the Cleveland Clinic, and it's recommending dietary changes for treating gout. And the particular thing that stood out to me, and this may be, I don't know how many of you do a whole bunch of cooking. I'm vegetarian, so I don't do this. But when you add, often meals will have, you'll have a recipe and it'll say, okay, you're going to add anchovies. Anybody know why you add anchovies? Not fish oil, but so you guys know umami? Mm-hmm. This is a savory. Okay, so I'll say savory. So that's like a glutamate, but also you get a lot of savory from nucleotides. Okay, so you get nucleotides, right? You can have add MSG or something like that. But so nucleotides, something that has a lot of DNA in it. Sardines actually have a lot of DNA in it, and that gives you a savory taste. So people will often add anchovies to things. Okay. Do you guys remember what causes gout? It's uric acid, which is a purine, right? Which is a breakdown product of nucleotides. So that's one of the reasons that they say to avoid this. I don't know how much of you have beer and alcohol. I don't know how game meats may have a lot of nucleotides in them as well. But that's the reason that one is on the list. And then I'm going to just skip on the ones that you're supposed to eat. Okay, but we are going to talk about nucleotides today. We're going to look over the breakdown pathways, but really I don't expect you to know these pathways. If I gave you one, I'd expect some sort of interpretation. But the main thing I want you to know is nucleotides are mostly made from the carbons that are in amino acids. And really, I'm talking about the base part here mostly. Obviously, where do we get this from? This is ribose 5-phosphate. Where does that come from? Pentose phosphate pathway, right? So let's go ahead and pentose phosphate pathway products. All right, and we're also, so that's really the upshot here. We're going to look over these pathways. You should be able to take one of these, like I said, and kind of interpret it. But here's the pathway for making. This is going to be, so we're going to here. Are we making purine? Is this a purine or a pyrimidine? This is a purine. Okay. The pyrimidines are the small ones, the purines are the 2-ring ones. And so if we look here, we start off with ribose 5-phosphate. So this is coming from the pentose phosphate pathway. Go ahead and pentose phosphate pathway there. So it's coming from glucose then, right? Pentose phosphate pathway starts with glucose 6-phosphate. And as we go along here, we've got some ATPs being melted, but here's a glutamine going to a glutamate. So what do we get out of a glutamate? If we go from glutamine to glutamate, what do we lose in terms of the amino acid? The amine, right? On the end of the side chain, and there we go, we got an amine now, okay? We can come down, we've got a glycine that gets added. We've got, here we have another glutamine to glutamate. So we're building up this ring on the ribose 5-phosphate, and so we're continuing to add the pieces we need. So we build one ring, we build the next ring. Here is one example where we don't have the addition of it or using amino acid to get a carbon. So here we've got can you see that? It's a bicarbonate, right? So we have bicarbonate, probably a what cofactor involved here? Which cofactor? The biotin, right? Remember, biotin's good for taking bicarbonates. Here we've got an aspartate, and the whole aspartate gets added. Just want to kind of give you a feel for what's going on here. But so we're building this up with either single atoms from, or not many atoms, small groups from amino acids, or actually there are places where we're adding a whole amino acid. We add a whole glycine. We add a whole aspartate. Yes.
So what is this?
This is the pathway of, you're going to take a ribose 5-phosphate, you're going to build a purine onto it.
Okay, so
You're building a purine. And just to put it in terms of the context, so we're going to make an IMP, and then after this, there's going to be a pathway that's going to take this to make this into an AMP or a GMP. Okay, so that IMP there, that's going to make our AMP or our GMP. And that concept of where these carbons come from is shown here, not as a pathway, but just as here, I've got a purine. Where do these various atoms come from? So we've got the aspartate, what's left of that aspartate after it's gone in. Here we've got carbon dioxide is going to bring this in. Here we have what's this guy? Formal tetrahydrofolate, right? So that's one of those tetrahydrofolates. So here we've got a carbon. Well, where did that carbon come from? Probably. We're getting them off of amino acids, right? Remember, we've got that serine to glycine bit. You don't need to remember that, but we've got that sort of thing. So the upshot here is I wanted to show you the pathway. I want you to have kind of that understanding that we're just taking pieces of or whole amino acids, and there's a biochemistry to make this it's a really complex sort of molecule when you think about what our amino acids look like and what that looks like. And we see the same thing when we look at pyrimidines. So pyrimidines are the single ring ones. So here's the overall path. And obviously they're not simple paths. Okay, but here what we've got is, oh look, we got a carbonyl phosphate. Where do we see that? That's the beginning of the urea cycle, right? We've got that. We're going to combine it with an aspartic acid. We'll go ahead and follow along here. Get some NADH. I'm looking for some more amino acids here. Not so much. This isn't as complicated. We've got a glutamine going in up there. Okay. This to make a pyrimidine. So, and our pyrimidines are our T's, our U's, and our C's. Okay. And so we can look over here. Here we've got a glutamine coming in. Okay. So you've got a lot of various places. Here we have 5,10 methylene tetrahydrofolate. And not, we're doing 5,10-methynyl tetrahydrofolate, but in lab, but that's what gets made. But so we've got, here we've got a 1 carbon unit coming in, and here, that 1 carbon unit coming in is going to take us from UMP to TMP, right? The difference between a U and a T, let me go ahead and draw them for you. That's a U, perhaps with some nitrogens. So that's a U, and then we think about a T, so a uracil or a thymine is that. Let's see if I can get them all on the screen at the same time. It's just one methyl group, okay? But it's really important. So we see this in RNA, we see this in DNA. It's really important to keep your DNA pristine, to not have any changes occur to it, right? There are mutations that happen, but they're very rare. Most times you have a mutation, it's bad, right? It's either neutral or bad. Very rarely do you get superpowers. And so what we've got here is this methyl group that gets added is very important for DNA repair. And I'll talk about why when we get to that, which is it's after this test. But also in lab, we've talked about, so I talked about 5,10-methynyl tetrahydrofolate being used at, no, not 5,10, 5-formyl tetrahydrofolate being used as a chemotherapeutic, right? We've talked about it being used against cancer. And one of the ways that it gets used, one of the ways it gets used is right here. All right, so you add it, you have so 5-formyl tetrahydrofolate. Yeah, that's what we're using as our substrate for our enzyme. And we'll go a couple reactions here. This is our enzyme MTHFS. That's the enzyme we're using in lab. We've got another step here. This comes in and helps. One of the ways that you do this is you actually add something that's toxic when this reaction happens. Okay, it's a floral uracil. And you add your 5-formyl tetrahydrofolate, and it actually helps this reaction happen more, and you get a bunch of TMPs that are defective out of it. And that's how that works, okay? Does that make sense, what we're talking about? So in this case, it's that you're adding 5-formyl tetrahydrofolate, which is the substrate of MTHFS. It's that storage form of folate. And you're helping this reaction to run because you've got floral uracil that actually messes things up for cells that end up incorporating it. Okay. And what do we know about cancer cells? They're fast. Yeah, they're fast-growing cells. They have to make lots of DNA. So you give them some things that when they make DNA, it kills them.
So what is the toxic part?
Yeah, it's not here. It's a floral uracil.
So where's the floral uracil?
That's not. It's available. It's this. It takes this place.
Okay.
That makes sense? Yeah, sort of. I don't know the structure of the floral uracil where the floral is off the top of my head. I'm not going to guess. I know where I want to put it, but I'm not going to guess.
So, is that something you want us
No.
to
Out of this, you need to know that we get most of our carbons for building up these bases come from amino acids, right? We're going to make it. If we're going to make a nucleotide, we're going to make it by taking the ribose 5-phosphate from the pentose phosphate pathway, right, from way back at the beginning of the semester, and then we take a bunch of amino acids and take parts of them, more more or less parts of them get built up to make the nucleotide bases. Okay. And we also have seen carbon dioxide be used to make some of that. Yes?
Can you go back up to the purine level?
Yeah. You want the pathway or the ending structure? This?
Yes, thank you.
Okay. Any questions?
Will you be able to draw that?
Not yet. You were supposed to be able to draw it last semester, right? And I'm not sure whether I require it again when we get into nucleotides. Okay. Probably not, because I haven't required you to know your amino acids either, right? Mostly. I mean, you need to know things like glutamate and glutamine, right? Because you've got a couple reactions where you have to draw it. Okay. What we're going to do now, any questions before I move on? We're going to talk about metabolism at a larger scale here. We're going to look at organ level things of what's going on. And what we're going to talk about is, this is really about compartmentalization by organ. So different organs can do different things. We talked about this before in the context of, your liver's doing a whole bunch of work, right? In terms of when we think about a lot of these anabolic pathways, we think about gluconeogenesis, okay? We think about fatty acid synthesis. These things are happening in your liver. We think about your muscles as the contrast to that. Your muscles aren't doing any of that stuff. They are taking in the products, they're breaking them down, they're running so they can contract, but your muscles don't build up. They don't make glucose, they don't make fats, okay? And so I want to look at some different tissues here. And so you can stop if you don't want gluconeogenesis to run at the same time as glycolysis. Well, one way to not have gluconeogenesis running at the same time as glycolysis is to not even have the enzymes present in that organ, the cells of that organ. Okay. All right. So, but let's go ahead and we're going to look at here. It's a little bit blurry, but what we're going to do is look at various things that go on. So the liver is going to do certain things depending on the state it's in or the state of the body. All right. So here, if we look at the liver in terms of what we'll call the fed state, right? I just ate an apple, right? And so my body thinks after eating the apple and the coffees I drank and the banana I had earlier that I've got enough sugar around, right? I've got more sugar than I need. What it's going to do is it's going to take in that glucose, okay, and it's going to produce glycogen, right? It's also going to take in the glucose and it's going to be okay for it to use that glucose. Remember, your liver provides glucose, it's maintaining your glucose blood levels. So it's okay if there's excess glucose around for it to use it, okay? And then it can for its own purposes, it's also okay for it to break it down and make acetyl-CoA and make fats, and then it will export those fats, and those fats will go and be stored in adipose tissue. Make sense? Okay. So if I was to tell you this is what's going on in the liver, right? What hormone would be present if I was in the fed state? Insulin. All right. So insulin is going to signal that I've got excess glucose in my blood and the liver needs to take it up and do stuff with it. All right. Fasted state. What's going to happen then? Well, in the fasted state, various things are going to get broken down by the liver, and it depends on kind of some other conditions going on. But so fasted state, I don't have enough glucose around. All right. And so my liver stores glycogen, so it can break down that glycogen to make glucose. The liver can also break down amino acids. Now, it'll have some amino acids of its own, but really I want those amino acids are going to be coming into the liver. And you see that down where it has the fed and fasted state. Those aren't stored there. Amino acids are stored mostly in your muscles. Right? So if I'm really in a fasted state, in starvation, your muscles are a source of energy, and you're going to break down muscles to make amino acids. And you're also turning over muscle all the time. Okay, most of the stuff the muscle you turn over gets built back into muscle, but I think it's like 5% of it will actually just go to the liver and be broken down. And so you can break down amino acids and make glucose from them. If I can break down an amino acid, make glucose from it, what do we say about that amino acid? We have a name for that.
Keep, like,
Glucogenic,
Yeah.
right? So that would be a glucogenic amino acid. It's not shown here, but we also have amino acids that we can break down and make acetyl-CoA, and we call those what?
ketogenic.
Ketogenic, right? So let's go ahead and draw an arrow on here. Okay. Now, if I don't have a whole lot of glucose around, if my glycogen stores are gone, my liver still needs to make some sort of fuel. My heart wants glucose if it can get it. My brain runs on glucose. Okay, so does yours. So my liver has to do something to compensate. So it's going to break down fats, right? It's going to be doing this anyway, but it's going to break down fats. So if I can't get enough glucose out, it's going to make ketone bodies. And we talked about ketone bodies, and those go out into the blood and go off to the muscles and the brain and other tissues. All right, whether down here we have whether or not we're in the fasted state or the fed state, we're turning over amino acids, right? So we're always turning over amino acids, and we might take in too many amino acids in our diet, take in too much protein, and we need to get rid of the excess nitrogen. All right, so that nitrogen is going to come in as amino acids to the liver. It's going to come in as alanine. All right, we'll see that in a minute. And then you take the amine off of that, and then that goes into the urea cycle to make urea. We just talked about that. And then urea will go off, and eventually it's going to go to your kidneys. Also, so we've talked about last semester, you isolated an enzyme from chicken breast that made lactate, right? So when you don't have enough oxygen around to fully combust the glucose, right, to fully oxidize the glucose, then what happens is you make lactate so you can regenerate the NAD plus and you can keep glycolysis going a little bit longer. But the lactate, once you're resting, or even while you're still working, although you're going to outrun this, you're going to overwork the system. Okay, you're going to run out of it anyway, essentially. But what happens is that lactate, the muscles make that lactate. The muscles aren't going to build that back up to glucose because they don't do that. That's not their job. So the lactate's going to go to your liver. Your liver's going to take that lactate and build it back up into glucose. And then it's going to dump the glucose back in the blood for your muscles, your brain, etc.
That happens no matter what state.
No matter what you are, or no matter what, if I'm working out, I could be really well fed and I'm working out, and I'm going to break down amino acids as part of that.
What
Okay.
signals that?
And I'm going to be making lactate if I'm not getting enough oxygen in. So if I'm sprinting. What was that?
What would signal that? Like insulin signals in the fed state, glucagon signals in the fasted state.
Right. So in terms of, I'm not sure what the exact signals are, but the liver probably notices or recognizes it has lactate. So the liver actually, when we think about glucose, I don't know what happens, what the transporter is for lactate, but when we think about things like glucose, you've got a GLUT1 transporter here on the surface of the, well, let's say this is the liver cell now, okay, not the liver. But on the surface of a hepatocyte, you're going to have a transporter that keeps the liver at the same concentration of glucose as the blood, if that makes sense. So you got a transporter, it's going to just equilibrate the liver, and so the liver has this sensing for insulin, and insulin in terms of telling it to take in glucose, but it doesn't actively take in, it just puts more transporters out there. And then as the concentration of glucose rises in the liver, it gets used more. If that makes sense. pathways are also going to get activated. Hold on to that. Put a pin in that. And we're going to talk about that a little bit more, hopefully today. If not today, we won't talk about it. But I think we'll be able to get to that. Questions? We're not going to worry about the kidneys. It's not that they're not important, but we're going to focus on adipose tissue, muscles, and liver in terms of what they're doing. So in terms of what should you know here, I should be able to tell you this is going on, and you should be able to reason through the pathways, what should be happening, right? You're starving. What should the liver do in terms of its pathways? You just ate a big meal. What should the liver do? Okay. You just got scared by a bear, right? So this would be what adrenaline would do. But what's the proper response from the liver if you're running from a bear? Regardless of fed or fasted, what should you be doing? Yeah, you should be dumping energy out, right? You're not going to worry right now about storing things. Okay, adipose tissue. What's going to happen in the fed state, right? In the fed state, is adipose tissue going to store fats or release fats? Yeah, store, right? Don't even look at the figure, right? But in the fed state, so hopefully that's what I'm going to see in this figure, right? I haven't looked at it, but so we've got this. In that case, we've got glucose we've got fatty acids that are going to come in and be stored, right? Don't worry too much about the glucose coming in, okay? The glucose comes in. Adipose tissue doesn't do much metabolically, but this glucose is just a source of glycerol because the glycerol is the backbone for the triacylglycerols. Okay, focus on what the fatty acids are doing. That's where most of our energy is. How about the fasted state? Fasted state. Don't look at the figure. You're gonna release fats. Or you're gonna store fats. Release fats, right? Okay. And that's really all adipose tissue does. It's not going to build up fats. That's the liver's job. Okay, and it's not going to break down fats. It'll break them down in terms of breaking triacylglycerols to fatty acids, but it's not going to do beta oxidation. It's not going to do fatty acid synthesis. That's the liver's job. The adipose tissue just sits there, and its job is to be a big bag of fat. Okay. Let's go to our muscles. Okay, so we can talk about fed state. Okay, your muscles are going to have to do stuff, and they're mostly going to have to do things based on whether they're contracting or not. There's not going to be a lot of stuff going on here that's going to be dependent on whether you're fed or not. We're going to see this, right? But go ahead.
Would this be for skeletal muscles exclusively, or is it different? Like, would this be the same for like the muscles in your stomach versus your arms?
I would consider it to be the same for all of them, okay, in terms of what you're going here, because you're going to have an active state. It may be to certain different degrees, right? Mm-hmm. So if we think about, you can only lose so much muscle in terms of starvation, right? Yeah. And some tissues may be more important than others. So fed state, though, right? Muscles do store glycogen. They don't make their own glucose. They're not going to run gluconeogenesis to make this glucose. Okay, but they will take in glucose, and if there's excess glucose around, they will store that. So some of your glycogen is stored in your muscles, more is stored in the liver. For starvation, let's go ahead and take starvation down here. We talked about in starvation, your muscles become a source, an energy storage source. Your proteins will start breaking down, your muscles will start breaking down. They'll make it into amino acids, and those amino acids will be released out to the blood, and they'll come up here to the liver, and the liver will then take them and detoxify, right? And it's going to take these amino acids, and up here we're going to break them down to make glucose, right? And fatty acids. Let's actually talk about that. We're going to take, so here we have amino acids in starvation. We have amino acids coming in. Those amino acids can be made to glucose that gets released to where they're glucogenic. Where they're not glucogenic, they're going to become ketone bodies. I just said that it would make fatty acids, but starvation of liver is not making any fatty acids, right? It would just be doing beta oxidation. It might be making ketone bodies, but it's not going to make fatty acids. It's not going to do a anabolic pathway to build up fatty acids to dump back into the blood. All right, the active site for your muscle, active state for your muscles then, it's green. This is the sorts of things that are going on in the active state. Do these all make sense? We have, they're going to take in glucose that's been provided mostly by the liver. We've got glycogen stores in our muscles that are going to be broken down as well to make glucose. The glucose can be made to pyruvate. If you have enough oxygen, what's going to happen to that pyruvate?
You have enough oxygen.
Yeah, we're going to do the citric acid cycle. We're going to break that down to carbon dioxide, essentially, right? But if we don't have enough oxygen, what's going to happen is we're going to have to make lactate. And the lactate's going to go out to the blood, out to the liver, so the liver can build it back up to glucose. Yes?
How does muscle build?
How does muscle build more muscle?
Yeah.
There are muscle stem cells. So the special muscle stem cells that muscle, when you use it, it breaks, it actually tears muscle, and then that activates the stem cells to build more. Okay, you're in hex, so you may have a better explanation of that. But,
I also have a question about like pyruvate to alanine, so like beta alanine is used a lot in like pre-workout.
so part of what was
Is
it?
there a correlation between like pyruvate going to alanine and like beta alanine?
Beta
It's
alanine?
it's an ingredient in pre-workout, so I didn't know if that
Oh.
has anything with like energy production.
I don't even know what beta alanine is. Is it different than?
It's the stuff that makes you itchy when you take pre-workout.
It makes you itchy when you take it with pre-workout?
Yeah.
I'd have to read about beta alanine. I haven't heard of it before.
Okay.
So, but people have you take it because it's supposed to help you not break down as much muscle, or in terms of you've got the fuel right there to immediately build the
It
muscle.
helps you with your recovery time, so you actually see like an increased like force production.
Right. Sorry, I haven't heard about beta alanine, but it makes sense, right? I mean, I don't know why. I have to read about it. I don't know why you would take that as opposed to something that was covered kind of all the amino acids better. But okay, so the alanine here is about, so I'm active, right? So I am tearing up muscle, some muscle being broken down, and the alanine is where we're going to take the muscle is going to take those amino acids, it's going to use it for fuel where it can, and it's going to take the amine from those broken down amino acids, and we'll talk about this in a little more detail here. It's going to use a transaminase to stick them on the pyruvate to make an alanine. The alanine is what goes to the liver. Okay. Alanine, that's how we're going to transfer those amines to the liver. And the reason why that happens, when this is active, you've got lots of pyruvate around, so you've got the materials to add that amine to. And then our we'll take in fatty acids and break them down to acetyl-CoA. We'll take in ketone bodies and break them down to acetyl-CoA to run through the citric acid cycle and the electron transport chain. Does that make sense? So we've got a whole bunch of pathways we've learned here. If I give you a tissue, liver, adipose tissue, or muscle, you should be able to tell me which ones are on and which ones are off, right? Glycolysis, gluconeogenesis, beta oxidation, fatty acid synthesis, right? Those are big things. We can talk about breaking down glycogen or building up glycogen. Those are the main ones you want to not miss. Yes.
You might have said this and I missed it, but when do ketone bodies get used to acetyl-CoA when there's just not enough sugar?
Right.
Okay.
So but your liver's going to make ketone bodies when you don't have enough sugar in your blood, or if you're diabetic, it can't see the sugar in your blood. Okay, so let's talk about this. I've talked about this already. This is the glucose alanine cycle. Whoops. Let's see, is it labeled below? It is not. Okay. So the glucose-alanine cycle. This is how we're getting those amines that are going to be we're going to make into urea from the muscles to the liver. And so what I want to do is correct this figure a little bit. So this is not actually, this is this transamination. Where is that? It's not an alpha. It doesn't come from an alpha amino acid directly. What does it come from? Glutamine, right? So this is glutamine. This is alpha ketoglutarate, right? And then what's over here? Here's our alpha amino acid and alpha keto acid, right? Okay, so the muscle's active here. We've got some muscle turnover, and so any of the 20 amino acids, well, except glutamate, right, is going to come, and we're going to get a transfer of its amine alpha ketoglutarate to make glutamate. Then glutamate is going to transfer that. Remember, glutamate is the one that's transferring everything, so we only have to have 19 or fewer enzymes to do this. All right, then we go back over to the liver, and now we're going to get this alanine. Once again, that's coming in as alanine. I'm trying to decide if I can neatly fix this here. Alanine is going to transfer its amine to alpha ketoglutarate, and alpha ketoglutarate is going to provide the amine to the citric acid cycle, not the urea cycle. So I'll go ahead and make this into glutamate, and then I'll do that. Just because I don't have room, but it'd be the same thing as over there, right? Alanine, and we're going to get that alanine becomes the pyruvate, right? Alanine becomes pyruvate because it's giving its amine to alpha ketoglutarate to make glutamine, then that goes over here.
Can you say that again?
So what's missing here in this figure? It's what was same thing that was missing over here.
Okay.
Okay, but I'm looking at it and I'm not sure how that's a little bit of a mess. I'm not sure how I draw it over here that it's not a complete mess. Alanine, over here we had some amino acid, gave its amine to alpha ketoglutarate to make glutamate. Glutamate gave it to alanine. Back here, alanine gives it back to a glutamate, different glutamate, and glutamate hands off that amine to the urea cycle, like we saw at the beginning of our urea cycle in that figure.
So it's directly the opposite, except for the urea.
It's the opposite over there. It's not a direct transfer of that amine over there.
So there's alanine.
Yes. Yes. And I don't care so much that you would be able to exactly draw this piece of this out in terms of testing this, but I don't want you to lose this concept that glutamate takes an amine from any amino acid and hands it to the next one. Glutamate's always this intermediary between those two. All right, but what happens here is we get this alanine made, right? We're essentially transferring a pyruvate with an amine. We're transferring that over here. This is how we get those amines to the liver. But the liver's then also going to build up that pyruvate back to glucose and dump it back into the blood, and that will go back out to the muscle. It's a glucose-alanine cycle. All right, we also have the lactic acid cycle. Okay, and so this is when we don't have enough oxygen around to completely oxidize this pyruvate. The pyruvate gets made to lactate, and then the lactate is going to get shipped out in the blood. It goes over to the liver. The liver will build it back up as the glucose and then dump it back out again. Okay, does that make sense? Why doesn't the muscle just turn that lactate back into glucose? Let's say it really wanted to. Let's say it really wanted to.
The enzymes aren't there.
The enzymes aren't there, right? We have compartmentalization of those enzymes, right? Muscles can't even, you can't run gluconeogenesis at the same time as glycolysis in the muscles because you don't even have the enzymes here. You do have both sets of enzymes in the liver, which is why when I talked about which of these pathways is going, I'm usually saying in the liver. Okay, you may not have noticed that, but I've been careful to say in the liver because there are only certain tissues this sort of thing can happen in. Kidneys can do lots of stuff as well, but we're not. Lots of similar stuff to what the liver does, but on a smaller scale. Questions? Glucosalinine cycle, lactic acid cycle. So, and these are examples of compartmentalization by organ. But I also, stepping one above these figures, also expect that I could give you a set of conditions in the body, and you could tell me what pathways were running in a specific organ. Okay. So you're between meals, okay? Not starvation. What pathways are active in your muscles if they are active? Bridget.
If we have like no oxygen, we're gonna break down the glycogen to glucose to pyruvate to alanine and fatty acids into acetyl-CoA.
Okay, but what pathways?
Oh, yeah, like glycolysis.
Glycolysis.
And yeah, beta oxidation.
Beta oxidation. What else?
The glucose alanine.
That's not a pathway, but I mean it sort of is a pathway, but you could say the glucosalinine, but what in the muscle? Give me some more pathways. Give me some more. Yeah. Those are the big ones. Glycogen breakdown. You could certainly say the citric acid cycle, right? You decide to say the electron transport chain if you wanted to. Okay.
Would you also say, like, I don't know, like the citric acid cycle and the urea cycle are very close together, would that also be reacting?
What was that?
The urea cycle?
You could think about the urea. You
But
might
not
add the
in the
urea
muscle,
cycle
not-
as well.
Not in the muscle.
Yeah, I mean, it's kind of always active at some baseline level. It's going to be extra active if starvation's going on, right? But this is not starvation so much. So if you said it, yeah, it's always got to be running at some level unless you're not taking any protein in, right? But if you're not taking much protein in. But starvation is definitely going to become upregulated because you're actively breaking down muscle for your protein. Okay, I want to talk about one other thing here. I may emphasize diabetes after the test, but I want to talk about enzymes. So enzymes can be present or absent, but also different enzymes with different activity profiles are present in different tissues. So for the same enzyme, or for enzymes that have the same function, they can have different activity profiles depending on the tissue they're in. All right, and this is an example of this. Just leave it. This hexokinase, where have you seen that before? Yeah, this is the first step of glycolysis. So is glucokinase.
Isn't that It's like called something different.
Glucokinase?
Yeah, is hexakinase the first step?
Let's say catalyzes. Okay, it's going to catalyze the first step of glycolysis. One of these is present in the muscle, and one of these is present in the liver. Okay, and this is your kind of normal blood glucose concentration. It's going to be held about there. Which is which? Which is which? Which one of those is muscle and which one of those is liver, and why? I'll give you a minute to think about it. It's a tough question. Bridget?
Would it be the red line is the liver and the blue line is the muscles? Or because it would take a second, you have to like wait for the liver to do the thing for the muscles to be able to do it, which is why it falls behind it. Also because hex, that very first step is used in other pathways, and the liver does more pathways than the muscles.
That's interesting. I like that. It's actually the other way around. But the way to think about this is that, so this one's going to be the muscle. I'm not the muscle, I'm sorry. This one's going to be the liver. And this one's going to be the muscle. Which one of these should respond to the level of glucose in the blood, right? If you look at this enzyme, the more glucose that's in the blood, the faster this enzyme goes, right? Which one should respond to how much glucose is in the blood? The liver or the muscle? The liver, because the liver is supposed to take it in. So what's going on is the liver is taking in glucose, and the more glucose it's in, the more it puts a phosphate on it, right? And then that'll keep it in the liver because it's charged. It can't go back through the membrane, and then it can do its job. Whereas muscles are fueled on the glucose, right? Muscles grab glucose, take it in, and they're not letting it go. So a muscle takes in glucose, it gets it stored into glycogen, or it uses it right away, and it's not going to put it back out into the blood. Does that make sense? So what we've got is the compartmentalization, or the roles of these tissues determine the activities of the enzyme. And remember, when I talked about the liver before, the liver is in equilibrium with the glucose in the blood. So glucose comes in, it goes out, it goes in, it goes out, so that this concentration of glucose in the blood is the same concentration of glucose in the liver. Whereas what the muscles are going to do is they're going to take in that glucose, they're going to immediately make it into glucose 6-phosphate, so there is no glucose in there. And that gives you a concentration gradient so the glucose comes in. If there's never any glucose present as glucose in the muscles, that's going to cause it to diffuse in. Questions? Okay. We will stop there for the exam.